I'm John Dirks from the Gairdner Foundation and the Canada Gairdner Awards. I want to welcome you all to the 2014 Canada Awardees Lectures, the 55th year that we've been involved in doing this. I've only been here for half of those. Today they have the opportunity to hear from all our 6 Canada Gairdner International Awardees. And we're privileged to have Dr. James Allison from Houston, Dr. Titia de Lange from New York, Dr. Harold Dvorak from Boston, Dr. Napoleon Ferrer from UCSD in San Diego, Sir Mark Feldman and Sir Ravinder Mani, both from London but now at Oxford. And then we have our Canada Gairdner Global Health Awardee, Professor Satoshi Umra from Tokyo, and our Canada Gairdner Whiteman Awardee, Dr. Salim Youssef from McMaster University in Hamilton. This year's winners showcase a broad range of new medical discoveries relating to cardiovascular disease, cancer, immunotherapy in particular, parasitic diseases. And sometimes we're asked about how basic science relates to eventual innovation and the economy. And I couldn't have a better example than this year's winners, in that almost every one of them has been involved in basic science that has yielded huge clinical individual patient benefit and has given if you trace in the background what has been the economic and industrial benefit and societal benefit, it's in probably about $100 billion. So it's, and all this has happened over a 30-year period. All this has happened almost undirected, and so that support of research and its return has been exemplified by our 8 awardees today. I enjoy getting to know each one of the awardees personally, and they've all traveled as part of the Gairdner National program, which is from Vancouver to St. John's. They've touched base at 20 universities and 20 cities, and they're probably getting a little weary, and we thank them for their efforts as in the quest that Gairdner has to promote science culture to young people. So now I'd like to thank our sponsors. They've been listed on the slide: the Governments of Canada and Ontario, the University Health Network, CHR, and the University of Toronto. I'd like to introduce Dr. Alison Buchan, Vice Dean of Research and International Relations in the Faculty of Medicine, University of Toronto, to preside over the morning. Alison.
Thank you, John. It is my pleasure to welcome all of you on behalf of Dean Catherine Whiteside of the Faculty of Medicine. Kathy is really upset she's not here. This is one of the highlights of the year. She unfortunately is in Hong Kong, so I get the honor of being able to welcome you on behalf of the University of Toronto and the Faculty of Medicine. As John mentioned, I'm the Vice Dean Research and International Relations, so it's very interesting today at the Canada Gairdner Laureate Lectures to see how widespread the origins of the lecturers are. They have come from all over the globe, but apparently united through 2 areas, one in the US and the other in the UK. So it is with great pleasure that I extend my personal congratulations to each one of you. So the overarching link in today's presentations are an interest in a singular topic, which is how do cells regulate themselves. The span is quite wide. We're going from the first laureate, who is going to talk about how telomeres are regulated to keep DNA intact and viable. And then we're going to hear from 2 pairs of investigators. One pair worked individually to understand how blood vessels originate, and which is termed angiogenesis. And in that process, discovered a growth factor, vascular endothelial growth factor, and how it works to control angiogenesis. The second pair actually worked as a team for years and actually became sirs together. So we have 2 knights speaking to us today. Their joint quest was to understand how you could use anti-TNF tumor necrosis factor therapy in immune disorders and they have been extraordinarily effective at understanding and developing treatments based on that work. So first, I'm delighted to introduce Professor de Lange. She is director of the Anderson Center for Cancer Therapy at the Rockefeller University, and she is being recognized for identifying a mechanism protecting telomeres. Professor Lange.
So it's a great honor and a great pleasure to be here. Thank you all for coming, and thank you to the University of Toronto for hosting us. I'm going to briefly discuss what we have learned about the end protection problem, but I will first introduce the subject and tell you a little bit about the medical relevance of the subject. So we have focused on telomeres, the ends of chromosomes that are essential for providing stability to the genetic material. The end protection problem is simply put like this. Eukaryotic cells have the ability to detect DNA damage in the genome, and that sets off a signaling pathway. Let me try to use the mouse. A signaling pathway that can lead to cell cycle arrest or to cell death. Obviously, this signaling pathway should not be activated at the natural ends of chromosomes, because then cells couldn't divide. In addition, cells have a mechanism to repair DNA breaks in their genome, and these DNA repair pathways, if activated at the natural ends of chromosomes, would lead to serious rearrangements in the genome. So telomeres have the task to allow cells to distinguish broken DNA ends from the natural ends of linear chromosomes. Now, this problem, the end protection problem, is relevant to a number of human disease states. It's a tumor, it plays a role as a tumor suppressor program. It leads to genome instability in cancer progression, and it's the cause of a series of human diseases called the telomeropathies. Now, the reason why the end protection problem becomes an issue in human cells is because of the way telomeres are maintained. So I'm sure you know that telomeric DNA is maintained by the enzyme telomerase, a reverse transcriptase that can add TTAG, the repeats, to chromosome ends. So as a consequence of the action of this enzyme, all the ends of chromosomes in human cells, but in fact in all vertebrates, look the same. They have long arrays of TTAG, the repeats, in double-stranded form, and a long single-stranded overhang at the 3' terminus. Now, interestingly, in the human system, not in many other mammals, but in the human system, telomerase is downregulated. It's the H3 component, the reverse transcriptase, that is switched off during early development, and as a consequence, the telomeric DNA is not maintained. In fact, telomeres shorten in human cells quite fast, and this is because every time a cell divides and the telomeres are replicated, this long overhang needs to be regenerated, and it's regenerated by nucleolytic attack on the 5' end of the chromosome. So, as a consequence, in our cells, while telomerase maintains the telomeres intact in the germline, there is gradual and progressive shortening of the telomeres during the divisions of normal somatic cells. And this also occurs in incipient tumor cells. And this pathway, this curious downregulation of telomerase, has been proposed to be a tumor suppressor pathway that provides a replicative barrier after about 50 population doublings. At that point, a few telomeres may bite the dust and become non-functional, and then DNA damage signaling pathways are activated that will arrest the cells and therefore prevent further tumor outgrowth. This was a theoretical consideration. Ron DePino has done a lot of work to model it in the mouse, but very recently, nature has provided us with the ultimate proof, because this model predicts that if the telomeres are too long in the germline, somatic cells start out with an excess of telomeric DNA, and it will take much more than 50 population doublings to reach this proliferation barrier. And so it's now clear that this is in fact the case because there are human diseases that are due to inappropriately long telomeres. In 2 papers in Nature Genetics this year, it was shown that due to a mutation in the shelterin complex that I'll discuss with you in a minute, there are families in which children are born with telomeres that are much longer than our normal length setting. And so it takes much more than 50 population doublings to induce this replication barrier. And indeed, in these families, you see that the patients are tumor prone. So this is a tumor suppressor pathway, and it works very well, apparently, in our system. But in cells that have lost P53 and RB, they become blind to the DNA damage signaling pathways that enforce this tumor suppressor pathway. At that stage, the telomeres continue to shorten as the cells divide, and now more and more telomeres become dysfunctional and start being attacked by DNA repair pathways that should not normally be activated at chromosome ends. And this leads to massive genome instability. And in fact, some of the genome instability in human cancers can be ascribed to loss of telomere function early during tumor development. Eventually, tumor cells need to reactivate telomerase because the state of genome instability due to telomere dysfunction is not conducive to proliferation. And it's become clear from the work of Levi Garraway and others that very often in tumors, telomerase is activated by mutations in the H3 promoter. So what comes out then is a cancer genome that's heavily rearranged, that is now able to maintain some stability because their telomeres are repaired. So telomeres that are too long are not good, but telomeres that are too short at birth are also not good. They give rise to a set of human diseases that were first identified due to the identification of dyskeratosis congenita. This is a rare inherited disorder that is caused by dysfunctional telomeres. The main symptom of this disease is progressive bone marrow failure, but when the patients are treated with bone marrow transplant, they develop lung fibrosis or liver cirrhosis later in life. And dyskeratosis congenita is the first of what is now known to be a whole spectrum of telomeropathies that are due to mutations either in the telomerase complex, the reverse transcriptase, the RNA, associated proteins, or in the shelterin complex and its associated proteins that I'll describe in a minute. This spectrum of diseases goes from diseases that are very low incidence, very severe and early onset, developmental abnormalities, to much more frequent occurring diseases with a milder phenotype that occur late in life. For instance, idiopathic lung fibrosis is now known to be to defect in telomere maintenance or telomere protection. So for these reasons, we are increasingly interested in this basic problem in biology, and that is how telomeres solve this end protection problem. And early on, we made the assumption that the end protection problem is not only solved by the presence of this DNA that is maintained by telomerase, but that the DNA can only function to protect the telomere ends by interaction with telomere-specific proteins. And so we set out to find such proteins, and more than 20 years ago, we put a lot of effort into the identification and purification and cloning of the first human telomeric protein, which we called TRF1. This is a double-stranded DNA binding protein that binds all along the length of the telomeric repeat array. We purified it from HeLa cells based on simple gel shift approaches, purified it to near homogeneity, sequenced it, and lo and behold, it was at telomeres. So once we had TRF1, we and others used TRF1 as a tool to get more telomeric proteins. We found a TRF1 homolog, or a paralog, TRF2, in the database. We discovered its interaction partner RAP1. Judy Campisi found TIN2 through a 2 hybrid screen with TRF1, and TIN2, as we showed, is a critical factor centrally to shelterin because it links TRF1 and TRF2 together, now allowing shelterin to recognize the double-stranded telomeric DNA with 4 independent DNA binding domains because both TRF1 and TRF2 are dimers. This anchors the complex on the telomeres. It binds all along the length of the telomeres and brings, through the agency of a TIN2-TPP1 interaction, the single-stranded binding proteins, POT1 proteins, to the telomere. These were discovered by Tom Check's lab based on homology to unicellular telomere proteins. Now, this complex is very abundant. It's constitutively expressed in all cells. It's essential, and it is highly conserved in vertebrates. So we can use the mouse and its genetics in order to try to figure out what this complex does at telomeres. Mice have the same telomeres we have. They're about 5-fold longer, but the same basic principle pertains. The only difference is that mice have 2 POT1 proteins, whereas we incorporate the function of both proteins in a single POT1. So we have used genetics in the mouse, particularly Cre-mediated deletion in mouse embryo fibroblasts, as a means to figure out the functions of this complex. And the complex has a number of functions. It controls telomerase-mediated telomere addition by using TPP1 to recruit H-TERT, the reverse transcriptase, to the telomeres, as was shown by Steve Artandi and Tom Check's lab. And it negatively regulates telomerase through the action of POT1, so telomerase will not make the telomeres too long, which, as I described to you, is not a good thing. And we don't know exactly how POT1 does this, but we're still working on this since we found a negative regulation some time ago. The complex also helps cells to duplicate the telomeric DNA with the conventional DNA replication machinery. The telomeric substrate is a nightmare for the DNA replication machinery, and therefore the fork, the replication fork, tends to stall in these sequences, and TRF1 has evolved to help the fork to get through. But most of our work focuses on the end protection problem, to prevent the activation of DNA damage signaling pathways and DNA repair pathways by the core components of the complex. So we recently were able to remove all shelterin components from telomeres. This was done genetically by Anja Sfeer in the lab, who deleted both TRF1 and TRF2 from cells. And since the complex is anchored at telomeres through these 2 double-stranded DNA binding proteins, the whole complex comes off the telomeres. And in that setting, one then can enumerate all the threats that exist and that alter telomeres if not protected. We discovered a few new pathways and rediscovered that telomeres are threatened by 2 DNA damage signaling pathways, the ATM kinase pathway and the ATR kinase pathway. These pathways have evolved to recognize double-stranded DNA ends and single-stranded DNA, both pathological in the genome, 3 DNA repair pathways, and inappropriate attack of the telomere by nucleases. So I'm going to focus first on TRF2, which takes care of 2 pathways. It represses classical non-homologous end joining at telomeres, a repair pathway that fuses DNA ends together through factors such as the Ku7080 heterodimer without any regard for the sequence present. And it threatens telomeres because it makes an inappropriate ligation of one telomere to another. In addition, TRF2 functions to repress the ATM kinase, which is the signaling pathway that responds to double-stranded breaks. And so we do all these experiments in the setting of conditional deletion of our favorite genes, and from mouse embryo fibroblasts, it works extremely well for shelterin. It's rapid, the cells don't die right away, so we can study what happens to the telomeres. And shelterin is designed by nature for these experiments because it is very compartmentalized, and different components can bind independently to the telomeric DNA, such that when we delete TRF2, RAP1 comes off the telomere and TRF2 is gone, but all the other components remain at the telomere, allowing us to parse the contribution of TRF2 to the end protection problem. Now what happens, I just want to give one example of data to the chromosomes, is that they start ligating together. So normally mouse chromosomes look like this with the telomeres free at the ends of the chromosomes. But when you delete TRF2, cells start ligating all the ends together and basically trying to convert the genome into one long DNA molecule, sometimes circularizing the chromosomes. So this is classical end joining, which is initiated by the Q7080 heterodimer that is activated when TRF2 is not present, but all the other proteins are still there. And in addition, we see this activation of the ATM kinase pathway. And this is interesting because this pathway, like classical end joining, is initiated by an end binding factor, the MRN complex, which recognizes in ways that are not fully understood double-stranded DNA ends. So how does TRF2, this simple DNA binding, double-stranded DNA binding protein, repress the activation of these two pathways? Well, our work suggests that it does so by using an architectural ploy in which it remodels the structure of the telomere such that there is a strand invasion of this single-stranded 3' overhang into the double-stranded part of the telomere. This invasion can occur anywhere, and it forms a loop-like structure that we first visualized by EM that tucks in the telomere end such that the end-loading factors that initiate these two pathways cannot recognize the telomere for what it really is, a DNA end. So recently, we've tried to test this model stringently. By EM, we knew that 15 to 40% of the telomeres could be shown in this structure, but we had no information on what happens when you delete TRF2. So we did an experiment in collaboration with Zhao Zhang, who uses storm imaging, one of the high-resolution imaging modalities that she developed, and we were able to visualize telomeres in storm imaging in a high-resolution way. But this was only possible after Uli Doksani developed a spreading technique in which the telomeres are relaxed and much of the proteins are removed. And using this technique, we saw the presence of little looped structures in the telomeric DNA. Now, to test the model, we needed to find a setting in which we get presumably linear telomeres, but where the joining doesn't occur. And so we decided to test the model by deleting TRF2 from cells that do not have this kinase. This kinase is actually needed for the joining reaction, and so this was an ideal setting for us to look in. So using that setting, we did the storm imaging, and in the controls, you see the T-loops when TRF2 is present, but when TRF2 is deleted from ATM-deficient cells, we convert the loop structures into linear molecules. These experiments can be scored. We've developed very stringent scoring modalities to look at the frequency of looped structures in different genetic settings. And you can see that both in a normal setting when TRF2 is deleted and all the telomeres fuse together, of course the T-loops disappear, but also when the telomeres are not fused together. So this is very consistent with the model in which TRF2 protects the telomere from classical end joining and ATM signaling. And importantly, none of the other sheltering components, when we deleted those from mouse cells, showed a change in T-loop frequency. So we believe then that TRF2 in the complex has the ability to avoid these two threatening pathways at telomeres simply by remodeling the telomere into an altered architecture where the telomere terminus is tucked away and hidden from the end-loading factors that initiate these two pathways. However, this model, or this ploy, is not sufficient to solve the whole end protection problem. There are other problems at chromosome ends. And as I said, we know there are 6 different pathways that can be activated. And for one more pathway, we have molecular insights how it's repressed. And that is the ATR signaling pathway. The ATR signaling pathway has evolved to take note of single-stranded DNA. And so it gets activated when single-stranded DNA in cells is covered by a very abundant single-stranded DNA binding protein called RPA. RPA covers the single-stranded DNA and then recruits and helps to activate the ATR kinase. And then the kinase phosphorylates checkpoint proteins that lead to cell cycle arrest and components of the chromatin nearby that recruit factors that then mediate the repair. Now, if you look at the telomere structure, either in the T-loop configuration or in a linear state, presumably a linear state will occur when telomeres replicate. Both are a good substrate for ATR signaling because there is sufficient single-stranded DNA to activate ATR through the agency of RPA. So how is this problem solved? Well, this problem is solved by the POT1 proteins that bind to the single-stranded DNA. They are required to keep the ATR kinase signaling pathway silenced at telomeres. When we delete the POT1 proteins, we see that ATR becomes activated. An example is given here, where we see the accumulation of one of the DNA damage response factors at telomeres co-localizing with telomeric DNA in interphase nuclei. And we can show that this is ATR dependent with shRNA and not dependent on ATM. We also know that under these conditions, we see the binding of RPA, the initiating the sensor in the ATR signaling pathway to telomeres. And this provided us with a clue because RPA is never seen at telomeres, or mostly not seen at telomeres, when the POT1 proteins are present. So we propose an exclusion model, a competitive model, by which POT1, through its ability to bind to the single-stranded DNA, will keep RPA from attaching to the telomeric single-stranded DNA and activating ATR. Now, this model is nice, but it has its limitations because RPA, in fact, has the same affinity for telomeric DNA as POT1, as we showed in careful biochemical experiments. And we did quantitative immunoblotting for RPA and POT1, and it's much more abundant, about 1,000-fold more abundant than RPA, than POT1. So this is not good for a competitive inhibition model. But POT1 has the great advantage of being tethered to the rest of the complex. So when it releases the telomeric DNA, as any DNA binding the protein will do, it has an off rate, it will be locally tethered, and therefore its on rate should be much higher than the on rate for RPA, which is not tethered. So the prediction of this model was that the repression of ATR signaling requires the tethering of POT1 to TPP1 and to TIN2, and thereby its linkage to the anchors of shelterin. And so we tested this model genetically using mouse knockouts to remove TPP1. We found ATR signaling. We removed TIN2. Again, ATR signaling is activated and RPA arrives at telomeres. And probably the best experiment is this one done recently by David Frascas in the lab, who used the TIN2 knockout to complement it with a TIN2 that can do everything TIN2 does, except for the binding to TPP1. So now there is a good core complex here, and TPP1 and POT1 are still present in the cell, but cannot arrive at telomeres. And again, there is activation of ATR signaling in telomeres. So what we have learned then from the work is that the end protection problem is a complex problem. 6 different pathways need to be blocked by shelterin, this simple 6 subunit protein complex binding to a 6 nucleotide telomeric repeat. 666. That's the message here. And we've learned that the complex is very compartmentalized. It's remarkable in its versatility because it blocks the different pathways through entirely different mechanisms. Now, we have not done yet. We think the T-loop model explains a lot at telomeres, and RPA exclusion by POT1 explains a lot as well. But there are 3 more pathways that we don't have insight yet how telomeres manage to repress them, and that's what we're working on right now. So I will stop there and acknowledge the current people in the lab that contributed to the most recent work. I have wonderful students and postdocs. I want to particularly highlight Uli Doksani, who did a formidable job in analyzing T-loops by storm imaging. This work was done in collaboration with Zhao Zhang at Harvard. I should also acknowledge our long-term collaborator, Jack Griffith, who does all the EM analysis. Much of the work I described also relied on Anja Spheer, who's now at NYU, their own lab, who did a magnificent job in creating shelterin-free telomeres, which required making mice with 4 genes knocked out. And I should acknowledge the generosity of people in the DNA damage response field who have provided us with many knockout mice to cross into, and the funding agencies who are still, even today, generous in funding my work. Thank you very much.